Chapter 8 Gene Cloning Introduction Molecular cloning is a technique for reproducing DNA fragments. It can be achieved by two different approaches cell based and polymerase chain reaction (PCR). In the cell based approach, a vector is required to carry the DNA fragment of interest into the host cell. Molecular cloning consists of several steps. Step 1 Isolation of plasmid DNA. Note that vector usually contains antibiotic resistance gene. Isolation of plasmid DNA and DNA fragment of interest. Digestion of the plasmid and DNA fragment with restriction enzyme to generate sticky ends. Number 3. Ligation of DNA fragment with the plasmid to form recombinant DNA. Step number 5. Transformation of a living host cell with a recombinant DNA plasmid and propagation of the transformed host cell to amplify the recombinant DNA. Step number 5. Selection of transformed host cell with antibiotic. Step number 6. Isolation and verification of cloned recombinant DNA. Plasmid biology. Plasmids are self-replicating extra-chromosomal DNA molecules found in virtually all bacterial species. In nature, plasmid occurs in exuberant profusion, varying in structure, size, mode of replication, number of copies per bacterial cell, ability to propagate in different bacteria, transferability between bacterial species, and perhaps most important in the traits they carry. Most prokaryotic plasmids are double-stranded circular DNA molecules. However, linear plasmids have been identified in both gram-positive and gram-negative bacteria. The size of plasmid varies widely from several kilopasses to hundreds of kilopasses. Replication of plasmids depends on host cell proteins, but also may require plasmid-encoded function. Plasmid replication may be synchronized with the bacterial cell cycle, resulting in a low number of plasmid molecules per bacterial cell, or independent of the host cell cycle, allowing the proliferation of hundreds of plasmid copies per cell. Some plasmids freely transfer their DNA across bacterial species. Some only transfer their DNA into bacteria of the same species, and some don't transfer their DNA at all. Plasmids carry genes that specify a wide variety of functions, including resistance to antibiotics, resistance to heavy metals, sensitivity to mutagens, resistance to antibiotic sensitivity or resistance to bacteriophage, production of restriction enzyme, production of rare amino acid, production of toxins, determination of virulence, catabolism of complicated organic molecules, ability to form symbiotic relationship, and the ability to transfer their DNA across kingdoms. All plasmid vectors contain three common features: a replicator, a selectable marker, and a cloning site. The replicator is a stretch of DNA that contains the site at which DNA replication begins, the organ of replication, and that also includes genes encoding whatever plasmid encoded RNAs and proteins are necessary for replication. The selectable marker necessary for the following and maintaining the presence of the plasmid in the cells is usually dominant, as it's usually a gene encoding resistance to some antibiotic. The cloning site is a restriction endonuclease cleavage site into which foreign DNA can be inserted without interfering with the plasmid's ability to replicate, or to confer with the selectable phenotype on its host. Replicators. One of the ways that replicators are classified is based on the number of plasmid molecules maintained per bacterial cell under some set of standard growth conditions, the so-called copy number of the plasmid. This book defines high copy number of plasmids are those which exist in more than or equal 20 copies per bacterial cell growth and lipid LB medium, and low copy number of plasmids are those which exist less than 20 copies per cell. High copy number of plasmids tend to be smaller than low copy number of plasmids. They are more commonly used in molecular biological techniques since it's easier to prepare large quantities of pure plasmid DNA from cells that bear them. Low copy number of plasmids are utilized when it's important to control gene dosage of a cloned sequence. For example, when the DNA sequence or the protein it encodes make the host organism sick. High copy number plasmids tend to be under relaxed control of replication. These relaxed plasmid initiate DNA replication in a process controlled by plasmid-encoded functions. And replication doesn't depend on unstable host replication initiation proteins synthesized at the start of bacterial cell cycle, because their replication depends only on the stable host enzymatic replication mechanism. The copy number of these plasmids can be greatly increased or amplified by treatment of plasmid containing cell with protein synthesis inhibitors such as chlorophenicol and spectinomycin. These proteins, sense inhibitors, prevent cells over-replication initiation proteins required for chromosomal replication, allowing the replication enzyme to be commanded for plasmid replication. High copy number plasmids don't usually have any mechanism to ensure correct segregation of the plasmid to the daughter cell. They rely on random assortment to partition at least one copy of the plasmid to each daughter. Low copy number plasmids are usually under restrained control. Initiation of replication of these plasmids depend on unstable protein synthesis at the start of bacterial cell cycle, and this is synchronized with the replication of the bacterial chromosome. As copy number decrease, random segregation of plasmid copies is not sufficient to ensure that each daughter cell acquire a copy of the plasmid. However, most low copy number of plasmid carry genes that guarantee their maintenance in the bacterial population. Stabilization loci that encode a mechanism for killing plasmid-free daughter cells and a system for efficiently resolving plasmid multimers so that they can be appropriately partitioned have been identified. In addition, cis-and-trans-actin partitioning loci that is thought to be constitute an active mechanism for disruption of plasmid copies to daughter cells have also been identified. Plasmid incompatibility. For experiments that require that more than one plasmid vector can be maintained in a bacterial cell at the same time, another critical feature of plasmid replicator is whether or not two plasmid replicators are compatible. Two plasmids are said to be incompatible with one another and hence belong to the same incompatibility group if they cannot stably coexist. Plasmids generally incompatible if they share any function required for the regulation of plasmid replication. For example, coli E1 and BMB1 derived plasmids are incompatible with one another but are compatible with B15A plasmids. The incompatibility of coli E1 and BMB1 plasmids are a consequence of two facts. First, that the plasmid DNA replication of these plasmids are negatively controlled by RNA1, which acts in trans on another plasmids with the same primer RNA. And second, that these plasmids lack mechanism to ensure that each plasmid in a cell replication once per bacterial cell per cell cycle. Therefore, if a cell that contains BMB1 derived plasmid is subsequently transformed with a coli E1 derived plasmid, cells selected to contain the coli E1 plasmid will usually have lost the BMB plasmid. Selectable marker. In order to guarantee that the plasmid vector is taken up and maintained in a bacterial cell, there must be a way to select the plasmid containing cell. Gene encoding resistant to antibiotics such as ampicillin, tetracycline, kanamycin, and chloramphenicol are the most common bacterial selectable markers for plasmid vectors. Only the bacterial cell containing the plasmid will grow on the selective medium. Another dominant selectable marker and is occasionally used is immunity to infection by phage lambda. In addition, recessive markers are sometimes used in selection. For example, Leo-B and Escherichia coli cannot grow in the absence of leucine, and selection for growth of the strains in the absence of leucine allow isolation of colonies transformed with a plasmid that contain a gene complements Leo-B. Another dominant selectable marker that is occasionally used is immunity to infection by phage lambda. In addition, recessive markers are sometimes used in plasmid selection. For example, Leo-B and Escherichia and E. coli cannot grow in the absence of leucine, and selection for growth of these strains in the absence of leucine allow isolation of colonies transformed with a plasmid that contain a gene that complements Leo-B. For experiments that involve introduction of a plasmid vector into system with another bacteria, other than bacteria such as yeast or mammalian cell, it's necessary to select for the presence of plasmid DNA in their hosts. Selectable markers for yeast and mammalian system are generally distant from those in E. coli. In cases where a vector is needed that will be used in E. coli and another host, it's necessary for plasmid vector to carry selectable marker for both hosts. Cloning site. Today's plasmid vector contain multiple cloning sites, MCS, or polylinker cloning regions that can include more than 20 tandemly arranged restriction endonuclease sites. The sites in the polylinker are almost always designed to be unique within the vector sequence so that cutting the vector with a restriction endonuclease in the polylinker and cloning foreign DNA into the site don't disrupt other critical features of the vector. The plethora of sites in the polylinker ensure that the appropriate enzyme site will be available for cloning most DNA fragments, provide unique reference restriction sites for rapid restriction mapping of the insert, and generally allow for a great idea of flexibility when manipulating the clone DNA. The sequences that directly flank the polylinker site are often useful for manipulation and analysis of inserted DNA. Most polylinker sites are flanked by a sequence for which they are commercially available complementary oligonucleotide, for example, M13 reverse and -20 and -40 primer that can be used primary for priming polymerase chain reaction PCR and DNA sequences reaction. Such primers are useful tools for amplification sequencing of any DNA fragment inserted into the polylinker. Some polylinkers are ported by 8 cutter restriction sites, such as NOT1. These sites occur infrequently in DNAs and thus allow for the easy accession of intact insert fragment from the plasmid vector. Expression vector structure. Over the past decade, the variety of host and vector systems for recombinant protein expression has increased dramatically. Researchers now select from among mammalian, insects, yeast, and prokaryotic hosts. The number of vectors available for use in these organisms continues to grow. With the increased availability of cDNAs and protein code and sequencing information, it's certain that these and that yet to be developed system will be important in the future. Despite the development of eukaryotic system, E. coli remains the most widely used host for recombinant protein expression. E. coli is easy to transform, grow quickly in simple media, and require inexpensive equipment for growth and storage. And in most cases, E. coli can be made to produce adequate amounts of protein suitable for intended replication. Vector for expression in E. coli contain at a minimum the following elements: a transcriptional promoter, a ribosome binding site, a translation initiation site, a selective marker antibiotic resistant, and origin of replication. In general, things that affect these can affect the level of protein expression. At a minimum, transcription parameters in E. coli consist of two DNA hexamers located in -35 and -10 relative to the transcriptional start sites. Together, these elements mediate binding of about 500 kDa multimeric complex of RNA polymerase. Suppliers of vector for expression has selected highly active and indicible parameter sequences, and this is usually little need to be concerned until a problem is encountered in expression. A strong parameter may be not the best for all situations. Overproduction of RNA may saturate translation machinery, and maximizing RNA synthesis may not be desirable or necessary. A weaker parameter may actually give higher steady-state level of soluble intact protein than one that's rapidly induced. Most parameters will have some background activity. Parameters regulated by the lactose operator or repressor will drive a small amount in transcription in the absence of added inducer, IPTG. To minimize this leakage, around 10% glucose can be added to the medium to repress the lactose induction pathway. The growth temperature can be reduced to 15 to 30 degrees Celsius, and a minimal medium can be contained to no trace amount of lactose can be used. Parameter leakage is the only problem with expressed proteins highly toxic to the cell. The tightly regulated T7 parameter has very low background due to the low level of T7 RNA polymerase made in the absence of inducer. It is specifically engineered host cells such as BL21. It has been estimated that the fold of induction of transcription in the T7-driven BET vector system is greater than 1000, while the magnitude of induction obtained the lac repressor regulated parameters is generally about 50-fold. Factors affect the level of translation. Translation can be affected by nucleotide adjacent to the ATG initiator codon, the amino acid residue immediately following the initiator, and secondary structure in the vicinity of the start site. Most commercially available vectors for expression use optimal ATG and Shine-Dalagrano sequences. Secondary structure in the mRNA contributed by the gene of interest can be prevent ribosome binding. In addition, the downstream box AAUC, AAUCAC, AAAGUG found after the initiator codon in many bacterial genes can be enhanced translation initiation. Conversion of the amino terminal sequence of the gene of interest to one that comes close to this consensus may improve the rate of translation of the mRNA. Methods. Alkaline lysis mini-preps. The alkaline lysis procedures is the most commonly used mini-prep. Plasmid DNA is prepared from small amounts of many different culture 1 to 24 of plasmid containing bacteria. Bacteria are lysed by treatment with a solution containing sodium dodecyl sulfate SDS and NaOH. SDS denature bacterial protein and NaOH denatures chromosomal and plasmid DNA. The mixture is neutralized with potassium acetate, causing covalently closed plasmid DNA to reanneal rapidly. Most of chromosomal DNA and bacterial protein precipitate, as does SDS, which forms a complex with potassium and or removed by centrifugation. The reanneal plasmid DNA from the supernatant is then concerned by ethanol precipitation. Boiling mini prep. Bacteria that contain plasmid DNA are broken open by treatment with lysosome. Treat on an anionic detergent and heat. The chromosomal DNA remain attached to the bacterial membrane and is built to the bottom of centrifuge tube during a brief span. Plasmid DNA is precipitated from the supernatant with isopropanol. This procedure is recommended for preparing small amounts of plasmid DNA from 1 to 24 cultures. It's extremely quick, but the quality of DNA produced is so lower than from the alkaline lysis mini prep. Storage of plasmid DNA. Plasmids can be maintained for a short period up to 1 month in bacteria strain simply by growing on selective plates and storing at 4 degrees Celsius. For permanent storage, bacterial harboring the plasmid should be grown in saturation in the presence of appropriate selective agent. An equal volume of more than 1 to 2 millibacteria. 2 milli of bacteria should be added to sterile 100% glycerol or DMSO-based solution and frozen at -70 in sterile vials. Cells taken from storage should again be grown on selective plates, and plasmid DNA should be checked by restriction analysis. Plasmid DNA can be stored in TA buffer at 4 degrees Celsius for several weeks, or preserved for several years by stored at -20 or -70 degrees Celsius. Most investigators prefer to store plasmid as frozen DNA due to the wide-held belief that plasmid stored in bacterial is sometimes lost and rearranged or accumulate insertion sequences and transposons during storage or on retrieval. Although such rearrangements certainly occur during storage of plasmids in bacterial and staph vials, we are unaware of any report rearrangements affecting plasmids stored in frozen cells. Introduction of plasmid DNA into cells. Number 1. Transformation using calcium chloride. Escherichia coli cells are grown to log phase. Cells are concentrated by centrifugation and resuspended in a solution containing calcium chloride. Exposure to calcium ion renders the cell able to take up DNA or components. Plasmid DNA is mixed with the cells and presumptively adheres to them. The mixture of DNA and cells is then heat shocked, which allow DNA to efficiently enter the cell. The cells are grown in non-selective media to allow synthesis of plasmid-encoded antibiotic-resistant proteins, then placed in antibiotic-containing medium to allow identification of plasmid-containing colonies. Materials: single colony of E. coli cells, LP medium, calcium chloride solution, ice-cold, LP plates containing ampicillin, plasmid DNA, chilled 50 ml polypropylene tubes, Pacman JS 5.2 router or equivalent, 42 degrees Celsius water bath. Number 8. Prepare competent cells. Step number 1. Inculate a single colony of E. coli cells into 50 ml LB medium. Grow overnight at 37 degrees Celsius with moderate shaking. Step number 2. Inculate 4 ml of the culture into 400 LB medium in a sterile 2-liter flask. Grow at 37 degrees Celsius, shaking and 250 RPM to an OD 590 of 0.375. Step number 3. Allocate culture into 50 ml pre-chilled sterile polypropylene tubes and leave the tubes on ice 5 to 10 minutes. Step number 4. Centrifuge cells 7 minutes and 1600 xg, 4 degrees Celsius. Allow centrifuge to decelerate without break. Step number 5. Pour off supernatant and resuspend each pellet in 10 ml ice-cold calcium chloride solution. Note that resuspension should be performed very gently and all cells kept on ice. Step number 6. Centrifuge cells 5 minutes at 1100 xg, 4 degrees Celsius. Discard supernatant and resuspend each pellets in 10 ml ice-cold calcium chloride solution. Keep resuspension cells on ice for 30 minutes. Step number 7. Centrifuge cells 5 minutes at 1100 xg, 4 degrees Celsius. Discard supernatant and resuspend each pellet in 2 ml ice-cold calcium chloride solution. Dispense cells into pre-chilled sterile polypropylene tubes, 250 milli freeze immediately at minus 70 degrees Celsius. B. ACES competency of cells. Step number 9, use 10 ng of PBr 322 to transform 100 milli of competent cells according to the steps provided below. Plate appropriate aldehydes 1, 10, and 25 of the transformant culture on LB and pestle plates and incubate at 37 degrees Celsius overnight. Step number 10, calculate the number of transformant colonies per aldehyde volume times 10 power 5. This is equal to the number of transformant per microgram of DNA. C. Transform competent cells. 11 aldehyde 10 ng of DNA in a volume of 10 to 25 milli into sterile 50 milli round bottom test tube placed on ice. Plasmid DNA should be directly from ligation reaction. When this is done, more DNA is usually used. However, if there is more than 1 microgram of DNA in the ligation reaction, and if the ligation reaction is from low gelling melting temperature agarose, it's wise to dilute the ligation mix. Step number 12, rapidly thaw competent cells by warming between hands and dispense 100 ml immediately into test tube containing DNA. Gently swirl tube to mix, then place on ice for 10 minutes. Step number 13, heat shock cells by placing tubes into 42 degrees Celsius water bath for 2 minutes. Step number 14, add 1 ml LP medium to each tube. Place each tube on a ruler, drum at 250 RPM for 1 hour at 37 degrees Celsius. Step number 13, heat shock cells by placing tubes into 42 degrees Celsius water bath for 2 minutes. Step number 14, add 1 ml LP medium to each tube. Place each tube on a ruler, drum at 250 RPM for 1 hour at 37 degrees Celsius. Step number 15, aldehydes of transformation cultured on LP, ampicillin, or other appropriate antibiotic containing plates. Step number 16, when plates are dry, incubate 12 to 16 at 37 degrees Celsius. Procedure 2, one-step preparation and transformation of competent cells. This procedure is considerably easier than basic protocol 1 because it eliminates the need of centrifugation, washing, heat shock, and long incubation period. Moreover, competent cells made by this simple procedure can be directly frozen at negative 70 degrees Celsius for long-term storage. Additional materials: 2 of transformation storage solution, ice-cold LP medium containing 20 ml glucose. Step number 1, dilute a fridge overnight culture of bacteria 1 to 100 into LB medium and incubate at 37 degrees Celsius until the cell reach an OD 600 of 0.3 to 0.4. Step number 2, add a volume of ice cold to time TSS equal to that of the cell suspension and gently mix on ice. For long-term storage, freeze small aldehydes of suspension in dry ethanol baths and store at negative 70 degrees Celsius to use frozen cells for transformation. Thaw slowly and then use immediately. Cells can also be used if battled by centrifugation 10 minutes at 1000 xg 4 degrees Celsius, and this may increase the frequency of transformation. Discard supernatant and resuspend cells pellet at 1/10 of original volume and 1 time TSS prepared by dilute 2 power 2 time TSS proceeds with transformation as step 3. Step 3, add 100 ml competent cells to 1 to 5 ml DNA, 0.1 to 100 ng, to an ice-cold polypropylene or gas tube, incubate 5 to 60 minutes at 4 degrees Celsius. As in the case for related procedures, the transformation frequency or as measured transformants gDNA is relatively constant at amounts of DNA less than 10 ng. However, the frequency decrease at highly concentration. The time of incubation at 4 degrees Celsius is relatively unimportant. Step number 4, add 0.9 ml LB medium containing 20 ml glucose and incubate 30 to 60 minutes at 37 degrees Celsius with mild shaking to allow expression of antibiotic-resistant genes. Select transformants of appropriate plates It is necessary to heat shock to the transformation mixture. The expected transformation frequency should range between 10 power 6 to 10 power 7 colonies slash microgram DNA. Procedure number 3, high efficiency transformation by electroporation. Electroporation with high voltage is currently the most efficient method for transforming E. coli with plasmid DNA. The procedure described may be used to transform freshly prepared cells or to transform cells that have been previously grown and frozen. With freshly grown cells, it routinely gives more than 10 power 9 bacterial transformants per microgram of input plasmid DNA. Materials: single colony of E. coli cells, Lb medium, H2O ice cold, 10% glycerol ice cold, SOS medium, Lb plates containing antibiotics, 1 liter centrifuge tube, 50 ml neuroproton polypropylene tube, and microcentrifuge tube, chilled ice cold, Beckman G 6M centrifuge, and Beckman GS 4.2 router and adapter for 50 ml neuroproton tubes. Electroporation apparatus with a pulse controller or 200 or 400 OHM resistor. Chilled electroporation cuvettes, 0.2 centimeter electrode gap. Prepare the cell. Step number 1, incubate a single colony of E. coli cells into 5 ml Lp medium and grow 5 hours to overnight at 37 degrees Celsius with moderate shaking. Step number 2, incubate 2.5 ml of the culture into 5 ml Lb medium, 500 ml Lb medium, and a sterile 2-liter flask. Grow at 37 degrees Celsius, shaking at 300 RPM to an OD 600 of less than 0.5 to 0.7. Step number 3, chill cells in ice water past 10 to 15 minutes and transfer to pre-chilled 1 liter centrifuged bottle. Step number 4, centrifuge cells for 20 minutes at 40 to 100 RPM in Beckman G6M 2 degrees Celsius. Step number 5, pour of supernatant and resuspend the pellet 5 ml ice cold water. Add 500 ml ice cold water and mix well. Centrifuge cells as in step 4. Pour off supernatant immediately and resuspend the pellet with swirling in remaining liquid. Step number 7, add 500 ml ice cold water, mix well, and centrifuge again as in step 4. Step number 9, pour off supernatant immediately and resuspend the pellet by swirling in remaining liquid. Step number 9, if fresh cells to be used in electroporation, place suspension in the pre-chilled narrow bottom 50 ml propylene tube and centrifuge 10 minutes at 4200 RPM in Beckman G 6M centrifuge with GS 4.2 router and adapter 2 degrees Celsius. Step number 9B, if frozen cells are to be used for electroporation, 40 ml ice cold, 10% glycerol to the cell and mix well. Centrifuge cells are as described in step 9A. Transform the cells. Step number 9, set the electroporation apparatus to 2.5 kilovolts and 25 micro F. Set the pulse controller to 200 or 400 ohms. Step number 11, add 5 bg to 0.5 microgram plasma DNA and 1 micro to tubes containing fresh or seaweed cells in ice. Mix it, trapping the tube or by swirling the cells with the pipette. Step number 12, transfer the DNA and cells into a cuvette that has been chilled 5 minutes on ice. Shake slightly to settle the cells to the bottom and wipe the ice in with water from the cuvette with a chem whip. Step number 13, place the cuvette into the sample chamber. Step number 14, apply the pulse by pushing the button or flipping the switch. Step number 15, remove the cuvette immediately, add 1 SOS medium and transfer a sterile culture tube with a positive pipette. Incubate 30 to 60 minutes with a moderate shaking at 37 degrees Celsius. Step number 16, a plate aliquot of transformation culture on LP plates containing antibiotics.